Good afternoon. Welcome to the SEBBM webinars. Today we are having a webinar organized by María Gómez. María is the coordinator of regulación de expresión génica group here in SEBBM, and she's from the SEBBM, and she's going to introduce the speakers he has brought today. Thank you, María.
Thank you, Patricia, and good afternoon, everyone. Thanks for joining today's seminar, and it's really my pleasure to introduce you to our speaker today. Is Leonardo Beccari from the SEBBM, Centro as I, different departments. And Leo is a young investigator. He's currently a Ramón y Cajal researcher here at the Institute, and he has a very interesting career. He started research he graduated in biology from the University of Malaga, and then did a PhD with Paola Bobolenta in I guess it was the Institute Cajal at that time.
Yes.
And then he did a fantastic work studying how transcription factors contribute to the dosage of transcription factors was an important developmental cue. Then he did a PhD, the postdoc with Denis Duboul at the University of Geneva. And then Leo did amazing work, including the pioneering use of gastroloids to study transcriptional programs, and 3D chromatin architecture, which is two of the things that I believe he will be telling us today. He did, as I said, very interesting contributions, which led him to obtain in 2020 a junior group leader position at the Institute of Neuromyogeny in Lyon, in France, and even got a permanent position at the INFERM there. But I don't know exactly why, but in 2022 he decided to was awarded the Ramón y Cajal, and then decided to come to the SEBBM, where we are very lucky to have him here. Because besides the fantastic contributions of Leo, he is also a super enthusiastic and helpful scientist here. So well, I leave the time and the microphone to Leo. Thank you.
Okay, so many thanks, Maria, for the very nice introduction. It's really a pleasure to be presenting the work here. I hope you hear me well by the microphone. And thanks to all of you that are connected. So the work that I'm going to present today, I'm going to share the screen in a minute, is really the work that I have initiated 3 years ago with the Ramón y Cajal Fellowship, meaning that still some parts of the work are still work in progress. So well, I hope you can enjoy the presentation, and every feedback is more than welcome. So I share the presentation. Yes, I hope you can see it. Yes, and then
yes.
Okay. I was trying to activate again the pointer, which before I managed to do, but now I don't see the option anymore. But well, so my group investigates the transcriptional mechanisms that control embryonic cortex development. And well, just to put a general introduction, the human embryonic cortex has really complex structures that depends on many different types of neurons, which generally can be grouped in 2 categories: glutamatergic, that are activatory, and GABAergic, that are inhibitory. But within these 2 big categories, in fact, there are huge diversity of neurons. and they are all generated during embryonic development, mainly from two niches. So the glutamatergic neuron arise from the dorsal part of the embryonic telencephalon, while the GABAergic neurons originate in the ventral portion of the telencephalon for a place named embryonic ganglionic eminence, and they have to migrate this long distance to integrate in the cortex. But doesn't matter in which place they are generated, they are produced through a similar process, which starts with the transformation of the pseudostratified neuroepithelium that is forming the wall of the neural tube into an epithelium of neural stem cell called radial glial cells. And these can, at the beginning, expand their number. And after approximately 10.5 in the mouse, they start the neurogenesis process, producing neuroblasts, either directly or, indirectly, via the generation of a more committed populations of precursors, which in general, they are called intermediate progenitors. Now we know that there are several types of these precursors, but in all cases, what this result is that these intermediate precursors then can further proliferate and also produce neuroblasts. And all these neuroblasts will then migrate to their final position within the cortex and differentiate into mature neurons. So as you can see, it's a rather complex and very dynamic process. And this control, it needs to be very tightly controlled by complex genes, complex network of developmental genes that give the identity, the ones particularly at the beginning, that give the identity to each region of the neural tube, and then during the neurogenesis process that control the timely differentiation of these precursors and their behavior and maturation of the neurons. So in our group, we address how this complex process is controlled by trying to understand how the expression of the genes that are involved in this network is regulated. And for that, we study them at three different levels: at the level of the enhancer code that controls the expression of these genes, at the level of chromatin organization, and at the level of the epigenetic control of promoter or enhancer activity. And while we have a sort of a fundamental interest in understanding this process, all these aspects have also important relationship with neurodevelopmental disorder, as mutations that affect each one of these processes can have a very severe consequences. And so to address these questions, we use brain organoid as a model system. And for those of you that may not be used to this model, I wanted just to briefly introduce them. So they are 3D aggregates that can be derived from human iPS or ES cells. And it always starts with the aggregation of these ES or iPS cells forming 3D aggregates that in principle are sort of indifferentiated. But then by exposing them to different combination of morphogen or morphogen inhibitors, in this case, inhibitors of BMP and TGF and Wnt, we provide the identity of the correct brain region that we want to reproduce, which in our case is the dorsal telencephalon. And this is manifested by the formation of this bright ring of cells in the outside of the organ. Then in the next phase, these organoid are these neuroepithelial cells are forced to proliferate, reproducing the very initial step of that correspond to the closure of the neural tube and the onset of the neurogenic process, in which there is this neuroepithelial to radial glial cell transition, and these radial glial cells start to amplify their number. And this results in the formation of these vesicles, where in each vesicles is sort of reproduce the neurogenic process that occur in vivo. What you see here is a light sheet microscopy of an organoid already more advanced, where you can see by different immunohistological markers the different population of neural precursors of neurons that are expected to have in vivo. Here you see that these precursors recapitulate quite well the organization that is described in vivo. Here, if we compare with a mouse embryonic cortex of approximately equivalent stage, and also the formation of the upper layer and deep layer cortical neurons. And so in this talk, I'm going to talk about two projects. The first one, which I would like to start with, talks about how the interplay between enhancer activity and TAD organization contribute to regulation of important neurodevelopmental genes. And this is the work that has been developed up to now mainly by Jorge Mañez Garcia, that is a wonderful PhD student that is doing the thesis with me, with the contribution of our lab technician Raquel Marco Ferreres. So just to put in the context, since the incoming of next generation sequencing, we have now plenty of tools to identify elements in the non-coding genome that may bear regulatory activity. In fact, for the human fetal cortex, this data set points to more than 50,000 regulatory sequences. And we also know that mutations in many of these sequences may have are associated with congenital disorders, but the activity, the precise function of each of these elements is not known, or is quite difficult to investigate, as well as it is to understand how mutations affect their activity. And the other important aspect is that these elements can be located very far from their target genes. And one concept that became very useful to understand how they interact, the gene-enhancer interaction works, it was the incoming of the concept of topologically associated domain, which were initially sort of described as a result of the high-C experiment, which are, here you see an example of this experiment, typically yield these heat maps in which interactions between two parts of the genome, here in yellow, for example, is represented by bin of different intensity, depending on the strength of the interaction. And this led to the observation that the vertebrate genome is compartmentalized at the 3D level in domains called TAD, for topologically associated domain, where DNA are more favored to interact together than with sequences outside. And so the work of many researchers, many groups got interested in this type of organization and showed that if we disrupt the organization of the TADs, and in particular of the boundaries that separate different TADs, this can lead to developmental gene deregulation and to pathological consequences. And while this is a concept that remains essentially true, and is a very important, was a very important breakthrough, over this year we have been observing how the more we look at this type of organization, the more we see that this TAD boundary, they do not have only an insulating function. For example, in some work they have been described that being at the border between two TADs is important to implement complex spatiotemporal expression patterns that genes and enhancer located near the boundary are more interact more easily, or that even they can jump the insulation of neighboring TADs, as well as that this organization may help TAD insulation. So it's what I want to convey with this slide is that the more that we look at the functional implication of this TAD organization, the more cases the more application we find. And in fact, different groups have reported that developmental genes are relatively often found at TAD boundaries. So to to learn more about what will be the functional implication of this organization, I got interested in the study of the human TBR2 gene, which is a transcription factor that plays an essential role in the cortical neurogenesis process. In fact, it is specifically expressed in this population of intermediate progenitors. And if you look at epigenetic data, genetic and conformational data, you can see that TBR2 is located precisely at the boundary between two large TADs, one of 700, almost 1 kilobase, and one of 500 kilobases. These two TADs, by epigenetic, display a lot of enhancer marks. And as far as I can tell, TBR2 is, in this context, is a little bit the only gene that has like a very specific function in neural development. The other are sort of constitutively expressed genes. So there were quite some chances that this regulatory element will be, in both cases, regulatory elements of TBR2. So, of course, we wanted to address the regulation of these genes using our brain organoid model. So the first that we did was to make sure that the organoid recapitulate the dynamic of TBR2 expression. And this is the case, as you can see in this qPCR and immunofluorescence analysis. And also by performing epigenetic analysis against different enhancer activation marks or chromatin accessibility, we could actually validate that many of the putative enhancers that were identified in the in vivo structure were also active in our organoids. And not only that, but if we perform for CSIC in our brain organoids, we can see how TBR2, that is this viewpoint in green, is able to contact both TADs. While doesn't matter which viewpoint we choose in either of the two TADs, the contact of that viewpoint, in this case, are two candidate enhancer of TBR2, you can see that the contact profile remain restrained to the TAD and entering a little bit in the TAD border, just enough to contact the TBR2 promoter. But this is quantified, in this graph. If we normalize the intensity of TBR2 contacts by the size of each gene desert, we see that the frequency distribution of TBR2 contact is almost the same between one TAD and the other, while for the other viewpoint is strongly segregated. And particularly, I want to call your attention in the fact that even in the TAD that I call here 5' because of its position, the number of contact is the contact are very sharp. They locate quite precisely in the vicinity of this element that we identify as putative enhancers by epigenetic profiling. And in the lab, we developed a transgenesis approach in the human iPS cells that allows us to integrate a reporter cassette carrying our candidate enhancer, and then with these iPS cells produce cortical organoid and see whether the enhancer drives the activity of the reporter in the organoid or not. And as you can see, for 3 of these elements that we have tested, they all show a very strong activity in our organoids, meaning that many of these elements are likely to be really enhanced. So the next step was to take a functional approach to understand the function of these 2 regulatory domains. And for that, we used CRISPR-Cas9 to delete either one or the other. And we were quite surprising to observe that when we do that, if we remove only one of these regulatory domains, and I want to highlight that we mainly remove the enhancers, but we don't remove any architectural element like CTCF binding sites, etc. So when we do this deletion independently, each one of them have a very little impact on TBR2 expression. In fact, we do not score any significant difference. This means that each of these domains, when removed individually, can be compensated by the activity of the other domain. In other words, that the 2 TAD redundantly regulate TBR2 expression. However, what was even more surprising to us is that if we now make a genomic inversion that changes the relative position of the TBR2 locus with respect of this enhancer, bringing it close to the SLC4A7 gene, what we have, is a very dramatic loss of TBR2 expression. In fact, we do not detect any TBR2, neither by qPCR, neither by immunofluorescence. So what this is telling you is that the position of TBR2 with respect of its enhancer and the organization in this TBR2 between these two tabs may actually be very relevant. And although we still do not have the final experiment to prove that, what we think in this arrangement, what happens is that we invert the polarity of the CTCFs binding site that are constituting the boundary, and this leads to the formation of an insulated domain which separates TBR2 from the other enhancer. Now, this, for us, it turned out to be quite relevant, not only to understand the function of these two tabs, but because it relates with another mutation that in this case was identified in patients, in a family of patients that have very severe brain developmental defect and that have a translocation that breaks the TBR2 regulatory landscape precisely as the point of the 3' tab. And it brings it into chromosome 10 in an inverted orientation. So we reproduce the exact same translocation using CRISPR in the human iPS cells, and we produce organoid from them. And we observe that, again, in this translocated, in the organoid with this translocation, we observe a significant reduction of about 50% of the level of TBR2, both in terms of mRNA and number of cells. And probably we think that this relates to the result of the inversion by the fact that in this translocation, again, we get that the new arrangement creates a new insulated domain that separates TBR2 from the enhancers that have come with it in the translocation. So basically, what I want to convey with this slide is that here we have an example where being located between two tabs is a mechanism that provides transcriptional robustness to the gene expression, and that altering this organization has even more impact than just disrupting a large fraction of regulatory elements. And with that, I want to skip to the second part of the talk to talk about the role of a chromatin remodeler. And this is the work of Daniel Felipe Cordero that did his master in the lab and is now starting the thesis, and again with the contribution of Raquel. So CHD2 is a chromatin remodeler that plays an important role in maintaining the target loci in an open state by exchanging the histone H3 by its variant H3.3, and it does that by recognizing the chromatin through its chromodomain and remodeling the nucleosome through the activity of the helicase domain. What is interesting is that in patients, these genes come often as candidate genes in patients that suffer epilepsy and other neurodevelopmental disorders. And different studies in mouse have shown that indeed it plays a role in cortical neurogenesis, but the precise mechanism is not well understood. So we initially investigated CHD2 occupancy and its correlation with histone mark, and this agrees with what is reported by other studies. It essentially correlates with active epigenetic marks, not with histone trimethylation, and it correlates with H3.3. However, there is a particularity of the human locus that, contrary to what happens with the mouse, it encodes for up to 5 different isoforms. And from the functional point of view, that this isoform can be categorized in 2 long isoforms that encode for all the domains of the protein, and other 3 short isoforms that encode for a truncated protein that only has the chromodomain. And what the prediction is that this short isoform can bind to the chromatin but lack remodeling activity. And the other data that is interesting is that patients with mutations that disrupt specifically the long isoforms tend to have much stronger and more precocious epileptic phenotype than patients with mutation that disrupt all the different isoforms of the gene. And this also goes associated with higher incidence of other behavioral or autistic problems. So our working hypothesis is that actually these isoforms are playing different roles, and mutations that disrupt all of them just lead to an overall decrease of all of them, but in an equilibrated manner, while mutations that disrupt only the long isoform lead to a debalance in the activity between short and long protein variants. So of course, this was the hypothesis, but need to be demonstrated. So we went to check which isoforms are expressed during neurogenesis, and we found that indeed is one of the long isoforms, the one that we call L2, and one of the short isoforms. And they become considerably upregulated during cortical neurogenesis. This we also demonstrated to happen at the protein level. And to make sure that the short isoform was being produced, we used CRISPR-Cas9 to insert a flag epitope specifically in the alternative exon 13, which is specific of the S2 isoform. And this is the validation, sorry. And we could see that already in iPS cell, this isoform is expressed and translated to the protein, So then we decided to explore what was happening with these different mutations reported in patients by generating mutant iPS cells that have a mutation called stop exon 5 that disrupt all the isoforms, or a mutation called delta exon 14-16 because it removes those exons that disrupt only the long isoforms. And with this iPS cell, we produce organoid and follow their growth and different parameters. So at the end of the aggregation phase, where I told you that there is no neural induction yet, there is no significant difference between either mutant or the wild type. But already at the end of the expansion phase, we start to see some differences. And intriguingly, the mutation that disrupt the long isoform, the 14-16, have a significant reduction in size, while the mutation that disrupt exon 5 do not have a significant difference. However, later on, in more mature organoids, both mutations seem to have a significant reduction in organoid size, meaning that probably they are altering the neurogenic process. So we went to this first time point where we first observed those mutations to have a different impact. And in fact, we could demonstrate that this difference in size is due to the fact that the exon 14-16 mutants display a reduced proliferation of the radial glial cells at this stage, while no difference was scored in exon 5. If we go a little bit later in the neurogenic process and we stain this organoid for marker of different neural precursor population, we see that neuroblasts, meaning newly produced neurons, are increased in both exon 5 and exon 14-16 mutant. But the intermediate progenitors, which are the landmark of this indirect neurogenesis, are only affected in the exon 14-16 mutants. So in few words, what we have is that we have a more pronounced effect on this long isoform specific mutant. And we think that this results in a stronger disruption of the pool of radial glial cells, which at the end leads, although at the beginning leads to increased neurogenesis, at the end will exhaust the pool and lead to a reduced neuron production. And to conclude, we started to investigate at the molecular level what is going on. So we did RNA-seq in these early stages organoid. And we see that, again, in embryoid bodies, there were very few genes affected. But at the expansion phase, there was a substantial fraction of genes that were downregulated in the exon 14-16 mutant, while the fraction of the regulated gene was much lower in the exon 5 mutant. Again, matching with the phenotype, with the morphological phenotype that we observed. And the genes specifically downregulated in this exon 14-16 mutant overlap largely with a different process of forebrain specification, neural precursors, proliferation, and neurogenesis or neuron differentiation. So, but what I want to highlight to conclude is that in this, the few genes that are downregulated in the exon 5 mutants are also downregulated in the other one, meaning what we are looking at is that a much more pronounced effect of one mutation with respect to the other, which sort of fits quite well with the phenotype described in the patients. And so our model that we are working on it to follow this track is that in normal conditions, the transition from IPS to neural precursors in our organoid models, there is a balanced activity of long and short isoforms that allows the long isoforms to open the chromatin of neurodevelopmental genes and allow their transcription. And in the exon 5 mutant, the proportion of long and short isoform, the amount of long and short isoform is decreased, but keeping the same proportion. And this results in a less efficient opening of the genes and their transcription. But, sorry, I'm finishing. In the exon 14-16, what we have is that apart from the reduction of the long isoform, we have a complete debalance of the short and long variants, and this has a much more dramatic effect on gene transcription. And with that, I just want to end by thanking all the lab members. I didn't have time to present you the work of Diego Martin Sanz and Eva Partner, that are also two amazing students of the lab. Eva is shared between the lab of Paola Bobolenta, so here at the SEBBM. And also I want to thank the collaborators, the founding, of course, and you for your attention. And I'm ready to take any question or comment.
Thank you very much, Leo. Very nice presentation. Let me check if there is any question from the audience. So far, it's not. So maybe I can start, unless you want to say something, Patricia, but I can start anyway if you want.
Yeah, well, I just have a question. It's because I have seen those beautiful organoids that you really get really beautiful ones that are like mimicking some parts of the brain, right? So how do you get them? Are they do you build those in your lab, or what kind of organoids are those?
So these are specifically cortical organoids because when we go to the induction phase, we supplement the induction media with BMP TGF inhibitors that give general neural identity, but also with Wnt inhibitors that promote anterior and dorsal fate. And yes, it's a homemade protocol, but essentially following the protocol of Lancaster et al. in
And they
2014.
are human cells, or are they mouse cells, or what kind of
These are
cells?
human iPS cells, yes.
Okay. I missed that part. Yeah,
And
they're beautiful.
we also produce ventral telencephalic organoid for the project, but or even assembloids. But yeah, for this particular project.
Really nice, and really nice to demonstrate your hypothesis. And yeah,
Thanks.
very nice. Thank you.
Yeah, sorry, I was muted. Yeah, I have two questions, Leo. One on the first part and then on the second. So on the first one, if I understood you correctly, what you think is, I mean, in both scenarios. So you have one scenario which is when you change the location of the TBR2 gene in the same that happened with patients, which is removing it into inside the TAD of the 3' site, and the other one is the one that you generated in the lab. That is not in patients.
No, the inversion is, let's say, an artificial construct.
Yeah, but in both cases, what you think is, do you think that you have the same kind of results? So you think that in both cases you are stopping the insulation. So now, if what you needed is either interaction with one TAD or the other, because you are now changing that architecture, that is not happening. Is that what you think? So you think?
So I think that because both in the inverted and in the translocated allele, what happens is that TBR2, the CTCF sites now get sort of in between TBR2 and the rest of the enhancers. And because in the two cases, those CTCF remain in convergent orientation with CTCF sites that are in the other part of the TAD boundary, this creates this insulated domain that prevent the enhancers to access to TBR2.
But it would prevent, in both cases, the ones that are in the 3' site and 3' TAD originally and the 5' TAD, because you saw also in the other experiments that you need either of the two, not both.
But the point is that maybe I can go. Do I still have the presentation active? If I can go to the slide again.
It's not active, Leo?
So here. Do you see the screen again?
Yes.
So let's see if I manage to go back. Sorry, there are quite some slides. Almost there. So we can stay here. So I think what is happening is that in the original configuration, the TAD 3', this the TAD 5', most of the CTCF are not in of one boundary or the other. I don't know if you see my pointer. This CTCF site and this CTCF site are not so much in convergent orientation, but instead they are in the same orientation. And my hypothesis, or our hypothesis, is that being in the same orientation, their insulation power is not as strong as being in convergent. So what happens when you invert or when you translocate is that now those CTCF become in convergent orientation with the CTCF of the other side, and this makes a stronger boundary. And now you have all the enhancer on one side of the boundary and TBR2 on the other side, and this cannot cross. But of course, for the moment, is just a speculation. We are in the process of doing high C to prove that.
Do you need to do high C? You cannot do the 4C.
Well, this is something that we are considering together with the lab of Viola Javier. And let's say we could, initially the idea was to do capture C, but at the end with the evolution of the sequencing cost, etc., I sort of trust the expertise of Viola in this. is more advantageous, in fact, to do the Hi-C, even if you get plenty of tags over the genome. But yeah, I mean, conceptually it will be the same. Is there because the cost of producing the probes for that region will be almost as much as just sequencing everything. But yes, that will be the idea. And I saw before there was a question in the chat.
Yes.
That I'm not sure if I can.
No, but I can read to you. So it's Eduardo Párraga Fernández, and he's asking you, how do you avoid necrosis in the organoids?
Yeah, so this in fact is it gave us quite some headache. So in general, we do organoid cultures that we collect around day 40. So they are not as advanced as, let's say, they are not so late organoids as other groups do, because for the processes that we are studying, most of the interesting thing is happening at that time. So already there, the necrosis score is not as big as one could think, but still at the beginning, and that's why some of the profile, they are not, let's say, perfect. We were having a lot of noise in our chip cat and tag 4C, precisely because of this necrotic core. But at the end, we implemented a protocol of dissociation and DNA treatment that sort of get rid of the nuclei. And now it seems to be working much better. So let's say we deal with the necrotic core at the stages that we are studying.
Okay. Maybe I can ask you something else.
Yeah, sure.
Maybe while we wait. So related to the CHD2 story, which is also very nice. Again, maybe you said it that I didn't understand you correctly, but so because both isoforms, so both proteins are present in the, and you said that during neurogenesis is increasing, the expression of both similarly. So mechanistically, what do you think they are happening? Because the short one only have the chromodomain,
Yes.
So you think that you need both, that you need different chromatin remodel activities and nuclei and helicase activities in different time points of development? How are you envisioning this?
In fact, so one hypothesis is that both isoforms will be sort of competing for the same loci in the genome. At the transcriptional level, the L2 and the S2 isoform are expressed almost equimolarly, but it's hard to say if at the protein level it's possible that there is a little bit more of the long isoform because I think this will make sense because the long isoform has much longer 3' UTR. So to me, it will make sense that its translation is more efficient than the short isoform, and so that in general the long 2 isoform would prevail. But I envision them to compete for the same site, and that under some circumstances the short isoform will be like an endogenous agonist of the long one. So what we want to do is that now with this iPS cell line where we have inserted an epitope in the short isoform and using an antibody specific for the long isoform to see if they really bind to the same genomic loci. If the hypothesis is true, I would expect that the short isoform will have a better correlation with repressive histone marks than the long isoform. But until I do not have an antibody that allows to, I do not perform this specific cat and tag experiment, I cannot give you an answer for sure.
But you don't know if it binds to chromatin on the short one.
It is predicted to, because it has the chromodomain.
Yeah.
And in principle, in vitro experiments, where people take only domains of the protein, at least in vitro, the chromodomain portion of THD2 has the ability to bind to the chromatin. So in principle, this is what we think that is happening. And well, in the iPS cells, you do see that with the flag epitope, you do see that the short isoform goes to the nucleus. So for me, it is in fact the main hypothesis is that the short is binding chromatin too.
Chromatin.
Yeah.
It's very interesting too. Yeah.
Okay. Perfect.
Okay.
So I don't know if there are more, because
it doesn't seem to. No. So okay, I think.
So many thanks.
Well, thanks to you, Leo, for this beautiful talk, and
thank you for both of you. Yeah, thank you, Maria, for bringing Leo, and thank you, Leo, for this beautiful talk.
Many thanks for the invitation.
If there are no more questions, I think we are ready to go.
Have a good afternoon.
Thank you very much.
Good afternoon. See you. Bye.
Cerrado. Vale, se ha ido Leo. Ya está cerrado el YouTube.
Vale, sí, se me olvidó de...